Hello, people.
Hello, teacher.
Let's see, what am I doing? What am I doing? What am I doing? My screen is weird. I see a big black screen with, oh, it's not black anymore. Now Claire is all over my screen. How do I get this back to a note? Not that it's not a good screen. It's just that it's not what I want. Not what I want. Oh, oh man. All right. I don't know what I'm doing. I know what you're saying. What else is new, Dr. Slish? What else is new? Oh, there we are. Okay. Now I'm back to normal. I can see everyone who's got their faces up. Hey, Lake, how's it going? Looking good this morning.
Thank you.
Okay. We don't have that many people. 23 out of 45. Why don't we just hang for a minute and get to know each other? Bring your faces up. Bailey, rocking the bangs, Bailey. Come on. I like it.
I don't even have bangs.
Oh, I'm sorry. I take it back. I don't like it. Oh, I'm a jerk. I know. Can't help it. Graylin. What kind of name is Graylin? I've been wondering about that. Your first name is interesting as well. Ms. Graylin. Oh, Mr. Sorry. First name, Graylin? Talking to you. He's laying down.
Yep,
Graylin, what's your first name?
Graylin,
What's your last name?
Larsh.
Larsh. What kind of name is that? Graylin. Is that Irish?
I think it's Gaelic.
Gaelic, okay. So I missed you by one day. We could have celebrated your name yesterday. Today you're just another schmo.
Yeah, yep.
Okay. Let's give it another minute or two. Sophia Martin. I don't think I've teased you in a while. How are you doing, Sophia? She's ignoring me.
I'm sorry, what?
How are you? I
I'm
haven't
good.
teased you
How
in a
are
while.
you?
I don't know. Hanging in. You know, doing my best. I think I'm suffering from what they call third quarter syndrome. Have you ever heard of this?
Not from a teacher, no.
Oh, okay. Well, they discovered this psychological thing for these people who go to like Antarctica. So you've got a winter station at Antarctica from, you know, for 9 months, and you're in the dark for 9 months with 19 other people. And when you get within, you get to leave, you suddenly start losing your shit. It's like, I feel like I'm supposed to get my vaccine in about a month and a half. And it's like, I just got to get out of the house. I've just, since October, I've gone to Price Chopper and Lowe's, and that's it. That's all I've done. So I'm losing it. I'm losing it. But the good news is, people, we're moving on to chapter 23, which is one of my favorites. The whole first half of this class, it's really important cell biology stuff, but it's not anything that I've like directly studied in the lab or anything like that. It's important information, but I don't find it that interesting, really. Second half, I think rocks. I love the second half of this class. So we're starting out with receptors and second messengers, which is basically pharmacology, which is what my degree is in. So, and it's interesting. Some of the things we talk about were just being figured out when I was in grad school. So it's old stuff. And then we're going to go from there from signal transduction, we go into, development. So if you're in my developmental class as well, the second half of this section is going to be pretty easy for you because all we talk about is Xenopus development, which is what we did in the last exam in developmental. So you got that. You can rest on that if you're in that class. And then the second exam, the fourth exam is going to be, again, kind of signal transduction leading to control of cell division, mitosis. And from there, we go into cancer, which is signal transduction, cell division, and all that too. So the second half of the class really rocks. And we're going to be going through course selection pretty soon. I'm giving a class next fall of the biology of cancer. So this is a good time to pay close attention, because a lot of that class is this chapter, except deeper and more detail and stuff. So signal transduction and how that controls cell division. So anyway, anywho, why don't we go ahead and get started? So I'd like to start today by going over our exam. I finished grading it this morning. I wanted to get it done before you came back. Oops, don't look. Which grades? Okay. So I did get it graded this morning. Oh, so I sent you that email the other day, and someone's like, what was that? It's like, one of the questions, well, we'll get to it. I'll talk when we get there. Exam 2. How do I do this now? I guess edit the exam. Matching. Oops, that's not what I want. Okay, question 1. Match the answer to the best definition. You can use each only once. So these are the kind of things that you would, hopefully the game helped you memorize all of this crap. So if you did the game and did well, you should have breezed through this. Using the gradient of one particle to move another, that's indirect active. Passive reversible uniporter. Passive reversible, that's got to be your glucose transporter, I'm guessing. Yep, glucose transporter. A passive reversible antiporter. This one. When a protein helps an ionic polar molecule across the membrane, that is facilitated.
You can see the question translation.
Oh, Jesus. I did this yesterday too. I'm sorry.
Yep.
I
was there.
you were there. Okay. Okay. So indirect active, glucose transporter, exchanger, facilitated. Okay. Pumps using ATP to produce a concentration gradient. That would be an ATP direct active. See, this would be right, but you can use each one only once. So you can't use it now. You have to use active transport. An ionic condition that causes cells to swell. Anyone who missed these two needs to go back to Bio 101. To shrink would be hypertonic. Energy dependent antiporter. That would be Mr. NCX. P-type pump. Now you got to use this one. Okay. So those are our correct answers, as far as I know. I could be wrong. I don't know why I say that, because I'm so very rarely wrong. Is that right, Bailey? I'm pretty much right all the time, right? 2 bonus points for Bailey.
For the most part.
Organelle for synthesizing lipids. That would be SIR, smooth endoplasmic reticulum. Amino acid that glycosylations in the rough ER. That would have to be asparagine. Process to degrade misfolded proteins. Rad. Synthesizing membrane proteins. Oh, that would be your rough ER. Glycosylations in the glucose. That would be serine, which looks like that, but it's not. It's this. Organelle for finishing proteins. That would be Miss Golgi. Enzyme that puts lipids on the opposite leaves. Flipase, floppase, scramblase. Moving from Golgi to rough ER. The retrograde, which makes the next answer enterograde. Okay. This is fun. I think I'm going to get an A in this class. Match the definition to the correct answer again. Transporter that is important for volume regulation in lymphocytes. This one is. It's the only one I. Energy conformation when NK binds the sodium. That's the low or E1. Protein used for calcium entry from outside the cells. Protein used for calcium entry from outside the cells. I would be expecting a channel. Oh, Caclium channel, my favorite channel. Transporter used muscle absorb glucose. Glucose transporter. And I specifically put because muscle cells are not, they don't need energy to bring glucose in. There's the sodium glucose transporter, which would be an answer down below, I believe. Let's see. Energy conformation binds potassium. That's the high energy, E2. Protein used for calcium release from the smooth ER. That would be our ryanodine receptor or IP3 receptor. Then we'll go with that. Transporter used by the kidneys to reabsorb glucose. Now we want to use our sodium glucose co-transporter because the kidneys are using energy to make sure they get all the glucose out of your urine. Enzyme that converts CO2 to that's carbonic anhydrase. Protein used to put calcium into the smoothie R, that would be your Circa. Okay, so again, I think a lot of these were in that game, so hopefully if you did that game, it helped you on this question. Matching, did I just do this one? 12? Did I do this one? Nope, okay. Retrieval tag, that would be Mr. Cadell. Side of the vesicles where vesicles enter, that's your cis-Golgi network. Liver enzymes for detoxification of the blood, cytochrome P450. Protein associated with targeting other proteins to lysosomes, that's your mannose phosphate receptor. Protein tag associated with retention is RXR. I always remember that because RER is the rough endoplasmic reticulum. RXR is the tag that keeps things in the rough ER. Small sphere of membrane used to move the proteins around. That's your transition vesicle. Side of the Golgi where enter for retrograde. That would be the trans-Golgi network. Main phospholipid of membranes, phosphatidylcholine. And last but not least, protein used to pass lipids to the mitochondria. That is phospholipid exchange protein. Right. A lymphocyte. Okay, now life gets complicated. These last four are the more complicated questions. So explain how a lymphocyte would regulate its volume if placed in a hypertonic solution. So a lot of people kind of gave me the Bio 101 answer. It's like if it's in a hypertonic solution, it's going to shrink because water comes out. Yeah, but that's not the question. The question was, once it shrinks, how does it get back up? And the way it goes back up to a large size is to bring more solute in, and then water follows the solute. So you've got your sodium hydrogen transporter, so hydrogen going out, sodium going in, your chloride bicarbonate transporter, chloride going in, bicarbonate going out. And you need to replace that hydrogen and bicarbonate, and you're doing that with carbonic anhydrase, water and CO2 combining to form carbonic acid, which then dissociates into a proton and bicarbonate. So that was the answer to number 1. That was kind of straight out of the notes, so it really wasn't that complicated. This is one that I'm not sure I put in the notes. I may have, but the information was definitely there. So this is why I sent you that email. If you look at the second sentence, it says list, bold and underlined, the proteins that are responsible. I would say half the people wrote me a paragraph this long. It's like, oh, now I got to read all this. The reason I put list was I wanted to be able to go from question to question going 1, 2, 3, 1, 2, 3, good. 1, 2, 3, good. But I had to read all these essays and I was cranky. Okay, calcium is approximately 1.8 outside the cell, and the inside is 0.5 micromolar. This is all 10 millimolar in the endoplasmic reticulum. This proteins are responsible for lowering calcium in the cell. So lowering the cytoplasmic calcium, you've got your plasma membrane calcium ATPase, your plasma membrane sodium calcium exchanger, and your Circa on the smooth ER, pumping calcium in there. That's all I wanted, just list those 3 proteins. And then proteins that allow things out into the cytoplasm, and that's plasma membrane calcium channels. Volschmann calcium channels 1, there are other ones. And then the calcium channels on the smooth ER. You've got ryanodine receptor and the IP3 receptor. So that was the answer for this question. Some people nailed it. They just kind of bang, 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 bang. Here's your answer. There probably people who have had me in class before, and they know how lazy I am. I don't want to read if I don't have to. Okay. Now you're a cell biologist. Okay, a lot of people didn't get what this question was asking. So I think a lot of people just didn't read it right. So let's read it together closely, because whenever there's a question this long, there's kind of a lot of distractors in there that are just kind of explaining background, but aren't really the question. This could have been any enzyme. I'm throwing in disulfide bridge enzyme just because it's something that happens in the rough ER. So as a cell biologist, you're studying an enzyme whose role is to form disulfide bridges in proteins while they are in the rough ER. Okay, so forget about disulfide, just think rough ER enzyme. You have mutated a cell line, which means the protein that we're looking at has got one amino acid change, most likely. You have a mutated cell line that is seriously deficient in disulfide bond formation compared to other cells. Okay, how do we interpret that? Well, the enzyme isn't where it needs to be, or it's not functional, one or the other. To help determine what the mutation is, you do immunocytochemistry on the mutated cell line and find the location of the disulfide forming enzyme. You find it's distributed throughout the rough ER, the Golgi, when it's normally restricted to the rough ER. Some's even being secreted from the cell. What's a possible explanation? So this question isn't about protein folding or disulfide bridges or anything. It's here's this protein that's supposed to be in the rough ER, but we're finding it in the Golgi. It's even being secreted. It's all over the place. What's going on? It's a targeting and trafficking question. So normally, this protein may normally have a retention tag on it, which means part of the sequence of the protein has got an RXR and it binds to a receptor there and gets stuck in the rough ER. It could have a retrieval tag, which means that it makes its way into the Golgi but comes right back to the Golgi afterward. And that would be your KDEL. Could also be a glycosylation site where glycosylations are added to it and that binds to a receptor and makes it stuck in the rough ER. If that amino acid's gone, it doesn't get glycosylated, it moves on forward. So the whole thing was about that, basically, trafficking and what would make a protein stay in. See, I could have asked the question that way and it would be so much easier. What would make a protein stay in the rough ER? And everybody go, oh, I know, a retention tag, But this is like, think about it. What are all the possibilities here? The three possible answers, and I'm still getting an A in this test, so I don't care. Okay, last question. Now this one's this is silly. This is kind of complicated, but I like this question a lot. And I would say 30–40% of you got it right. Suppose for a moment that in movie E.T., the scientists got their way and had a chance to dissect E.T. Does everybody know E.T.? This is an 80s. Sometimes my references are too old. So they found that E.T.'s stomach digested proteins by increasing pH in the lumen through a hydroxide ion pump. Okay, we don't have one of these, but apparently E.T. does. Potassium is also allowed to enter the lumen from inside the cells. You can kind of infer there, allowed to enter the lumen. It's not an active process, it's passive. Okay, so the net change is leaving KOH. How are you going to balance that out? A lot of people started the question like that. Net change is loss of potassium and hydroxide, and that needs to be balanced. And that's the question. How do you balance that using the transporters that we talked about in class? And there's a lot of ways to do this. One, you're losing potassium. So pump potassium back in with your sodium potassium ATPase. And then we need to we're lacking hydroxide, which means we have extra protons in there. So simplest way to fix that is you've got potassium high inside, and from the saliva mechanism, the salivary duct mechanism, we had a potassium proton antiporter. You could have potassium going out, potassium going in, and then the proton coming out. Yes, and that would bring the acid out of the cell. So both those mechanisms are bringing potassium in. Although I wonder if that transporter would work, because there'd be high potassium inside. That might not work. But you could do your sodium. No, you couldn't do that. So anyway, that's one way. The other way is use your sodium potassium ATPase to get potassium back in. And then to balance, this is what I was hoping people would do, and some people did, to balance the extra protons inside, use your chloride bicarbonate transporter, bring bicarbonate in. Bicarbonate then binds to the protons, and then carbonic anhydrate turns it into carbon dioxide and water, and the CO2 can just leave. So that's two ways to do it. There was another one that I was thinking of. I don't know, it may be in the answer to this question. But anyway, there are several answers here. None of them were yours because you all failed. I'm just kidding. Okay, let's just stop this. Okay, questions, comments? Hey, Barb. Barb came. Hi. Questions, comments, nothing? If you don't ask me a question, we're going to have to start learning stuff. I'm warning you. We're going to start learning. Okay, let's do it. Let's not be afraid of the future. Where is my PowerPoint? PowerPoint. Oh, me.
Slash, are you teaching any classes this summer?
This summer, just my drugs class. I always do that online. I think you've taken that already, haven't you?
Yeah, I did.
Take it again. I do get paid extra in the summer, so you would be helping my bottom line. You took it a second time. I'm guessing you're not going to do that. You're ignoring me. Okay. What are you, my wife? You're ignoring me. Terrible joke. I'm sorry. All right, here we go. Messengers and receptors. Just gonna pull up my notes so I can see them. Oh, so here's the thing. Another thing I wanted to tell you. These notes, I wrote these notes last year. Remember, this is our year anniversary of this COVID stuff, right? So this was the first set of notes that I wrote so I could do this class online. And I put a lot of work into this. So these notes are pretty good, A. And B, it's like every time you see like a highlighted thing, there's an animation that goes with it. That was my goal, was to put animations in with the notes. So as you're going along, you can always have that backup. I ran out of energy halfway through the semester. But I think the next time I go through this class online, or even when I do it in person, I'm going to do this some more because I think these notes are really nice. Okay, messengers and receptors. So this first picture is showing, like, I think anyway, I could be wrong, sperm entry into an egg. And when that happens, you get a spread of calcium from that point all around the cortex of the cell, with it being highest where the sperm entered. So it's like sperm entry activates the release of calcium, or calcium entry, and then that changes the whole membrane of the egg, and now you can't get another sperm in. It inhibits polyspermy from happening. You know, you get two sperm heads into the same egg, and then it's just not viable. So signal transduction. Yay, signal transduction. Okay, so I talk a lot about hormones and receptors. I just want to define that first. So a hormone is just a small molecule or a peptide, a small protein, and it's being released from one cell, and it's usually a different cell is responding to it. And receptor is always a protein. So the receptor is a protein that's got, you know, amino acids that specifically bind to that hormone. So you get a really tight interaction between the receptor and the hormone and the we call it ligand. Sometimes I call the hormone a ligand. When you're talking about the binding aspect of the equation, we always call the small molecule a ligand. Ligand just means something that sticks. Okay, hormone receptor, and sometimes I'll say ligand for hormone. All right, what kinds of hormone release are there? Slide 2. Usually when we think about hormone release, at least when I think about it, I think about endocrine release. So you've got a cell somewhere, like your adrenal medulla, releases epinephrine. Epinephrine goes into the extracellular fluid, gets taken up by capillaries. Capillaries go to veins, back to the heart, spreads all over your body. And then certain cells will respond to it. Not every cell does. The cell has to have a receptor on it. So for example, the corneal cells in your eye don't have receptors for epinephrine, so they don't respond to epinephrine. But the ones on your blood vessels do, so they do relax or contract or whatever they do. So that's endocrine, being released from one source, transported in the blood, and then the cells that have receptors will respond to it. Another one is paracrine. Para, we see that word a lot. It kind of means around or nearby or on either side of something. So paracrine is when one cell is releasing the hormone right onto the cell next door, and that cell has receptors and is responding to it. That's kind of what we're seeing here. This cell is releasing hormone and the cells next door are responding to it. So an example of this would be like, a fibroblast or a platelet. Let's say platelet. Platelet is a cell that responds to cell damage. So a platelet goes into a capillary and it finds damage. The capillaries burst open. It hits all these proteins it shouldn't see because now it's outside the blood system, and it gets activated and it releases PDGF, platelet-derived growth factor. All the cells in the neighborhood then will like wake up and start to divide to repair the damage. So that's a local kind of release of something. Also, the platelet may itself respond to PDGF. Fibroblasts do this too. They'll release fibroblast growth factor that'll affect all the cells around it, but also affect itself. And so if it's affecting itself, that's called autocrine. Okay, autocrine release. And then there's another one. Nope, that's not it. So there's another one called juxtacrine, is when Notch is a good example of a signal transduction pathway that does this. And if you're in the developmental class, we'll be doing Notch this week. Notch is when you've got two cells and one has got a hormone-like protein attached to its plasma membrane. The other has a receptor attached to it, and they come together and one touches the other. So it's like the hormone isn't released, it's two cells touching, and you've got kind of a hormone receptor interaction there, activating signal transduction in this cell. So an example of that we'll talk about, I think in this chapter, is apoptosis being stimulated by a natural T killer cell. So the T killer cell's got a ligand sticking out of its membrane called FAS, and you've got FAS receptors, and when the FAS ligand hits the FAS receptor, that cell gets signaled to go into apoptosis. So that's juxtacrine. Hormones bound, a protein that's bound to the surface of one cell touches another cell, and that activates signal transduction. Okay, hormone signaling. So how does the signal transduction happen? What is signal transduction? Depends on the hormone. There's so many different signal transduction mechanisms. It will make your head spin. At least that's my goal for this class. Make your head spin. One, probably the simplest one, and I never spend much time on this because I always assume it's obvious, so I'll move on quickly. And then people don't really get it. So I want to spend a little time here talking about this. One is if you've got a lipophilic hormone, and lipophilic means that it can get into the cell, right? It can diffuse out of the capillary and into the cell right through lipid membranes. And so the receptor for these is cytoplasmic. So here's your GR, what does that mean? Growth receptor? I don't know. The receptor is here in the cytosol. The steroid hormone comes in, binds the receptor, and that whole thing gets translocated to the nucleus, and then it binds to a control element in a gene. So an upstream regulatory control element, the protein binds to it, and that starts transcription from that gene. So that's your steroid hormone, your thyroid hormone. All your lipophilic hormones will work that way. Again, pretty simple mechanism, so I don't spend much time on it, but it's pretty important because this is seen a lot in there's a lot of signaling that is this. Retinoic acid we talked about in developmental if you're in that class. Retinoic acid is like this. Lipophilic binds to an intracellular receptor, which starts activation. Okay, enough of that because it's boring. Receptors, on the other hand. Oh, come on, you. What are you doing to me? My AV's failing me. Hydrophilic. So hydrophilic hormones, which most of them are, can't get into the cell. So because they can't get into the cell, their receptor is going to be cell surface. So it's a membrane protein in the cell surface will bind and respond to that hormone and then activate some signal transduction pathway. So these signal transduction pathways can be pretty long, but they're all kind of like straightforward in that this activates this, and this activates this, and this activates this. At least the way we present them here in class, that's the way it is. In reality, it's a lot more complicated, of course. You know, my high school chemistry teacher told me that teaching science is teaching a series of lies. The first thing you tell anybody about anything is like so oversimplified, it's not true. And then you say, oh yeah, but then there's this complication, get a little closer to the truth and a little closer to the truth, because the truth's always more complicated. Okay, so once you activate that signal transduction, a couple different things can happen. One is you could get changes in gene expression. We've got a whole group of hormones that'll do that. And other ones are more for cellular physiology. So like contraction, relaxation of a muscle, or, you know, causing a cell to secrete something. I mean, so it's fast and it's reversible. Whereas when you activate gene transcription, it takes a while to do gene transcription, so it's slow. And also you can totally change a cell and it never goes back. So it's not really reversible. For instance, if you stimulated the cell to divide, once it's divided into two, well, you can't reverse that. Now you got two cells, as opposed to contraction, relaxation of a cell. So it's just the same cell doing two different things. So once you activate the signal transduction, then based on the hormone and the receptor, the cell will either have some kind of a physiological reversible response or some permanent change by activating genes. Did I skip one? Nope. Okay. Slide 5. Is this slide 5? Holy cow. Okay, so there are a couple of different types of kind of general classes of receptor mechanisms. One, and probably the simplest, is just a ligand activated channel. So we talked about voltage activated channels already. This is a ligand activated channel. So like acetylcholine binds to the nicotinic receptor, it opens, sodium comes in, changes the membrane potential, and then the cell responds to that change in membrane potential. We saw that skeletal muscle contracting for that reason, or getting an action potential anyway, and then later contracting. So that is one type. It's just a ligand, very quick, very easy. We call these ionotropic channel receptors. Then you've got metabotropic receptors here, which has got a receptor that the hormone binds to it, it activates the receptor, and then you get activation of a series of enzymes, usually through G proteins. And the second messengers often used here are cyclic AMP and IP3. So we'll talk a lot about these two bad boys. And then you get a response from that. So these two types of mechanisms are both kind of that quick change mechanism, like it can change the physiology of the cell and then the cell can go back normally. And then this is the more long-term function here. This is your receptor, which is usually a kinase. The hormone binds to the receptor. You activate a whole series of intracellular kinases, and you get a response from that, which is turning different genes on. And that could cause the cell to divide or to differentiate into a different type of cell. So, so that's the third type. So this is kind of a broad overview of what, of the types of signals transduction in the cell. Now, what is that receptor doing? Good question. I'm glad you asked. Basically, the receptor is doing two important things. One is amplifying the signal. So here we're looking at this. This isn't really the way it works, where you've got one hormone binding to one receptor, activating one of these and coming up with one of these. The way it really works, oops, what am I doing? Is like this. You're getting a major amplification of the signal at many of the steps along the way. So we'll talk about cyclic AMP later. This is the cyclic AMP thing, but let's just look at the amplification involved here. So say you're in a stressful situation, your body releases epinephrine. Epinephrine is going to mobilize glucose stores, so you've got energy to respond to whatever it is. So epinephrine gets released from your adrenal medulla into the bloodstream, all over the body. The liver cells pick it up, and the epinephrine binds to the receptor. So you've got one molecule of epinephrine binding to one receptor, one-to-one interaction. That's what I call stoichiometric. That receptor then can activate 100 different G proteins, which is the next step along the way. So you've got amplification here 100-fold. And then each of those G proteins then will activate one adenyl cyclase, which is the effector organ. We'll talk about that, or effector molecule. We'll talk about that later. But that's a one-to-one, one G protein activating one adenyl cyclase. That's not amplification. And then that one adenyl cyclase that is active will make 100 molecules of cyclic AMP. So now we've amplified the signal another 100-fold. We're up to 10,000. Each of those then will bind to one kinase, one-to-one. That's not amplification. And then each kinase will phosphorylate 10 of these phosphorylase kinase enzymes. So now we've got another amplification, 10 to the 5th. And then each of those will phosphorylate 10 of these. So you've got another amplification. And then each of these will break down 100 glucose, glycogen, take 100 glucoses off glycogen. And so you get that release of that glucose 1-phosphate. So basically, what is that, 100 million fold? So you start with one epinephrine molecule, you end up with 100 million glucoses being released. That's the amplification of the signal that we see. And that's true of every pathway we'll talk about. And that's, you want to keep that in your mind. A lot of times we'll go through it when we do the cyclic AMP and IP3 mechanisms. But keep in mind that every step along the way, you have the possibility of amplifying the signal. So I always draw it like it's this, does this, does this, this, this. And it looks like a one-to-one pathway, but there are amplifications in there that really build things up. So that's one thing it does. And the other thing, oh, let's not get into that yet. And the other thing is to transduce the signal. It's taking the signal from this extracellular small molecule, and without the receptor, the cell wouldn't respond to it. But if he's got the receptor, then that brings the message from extracellular signal to an intracellular signal that the cell does respond to. So cell can't respond to epinephrine, but it can respond to cyclic AMP. So the receptor mechanism connects the first messenger to the second messenger, cyclic AMP. Transduction. Okay. How long is this class? Oh, we still have a half hour. I need to talk slower. I will talk slower. Okay. All right. So, oh, this might work. Hang on. Let's see if this works. No, this is going to work. No, this isn't going to work. Never mind. All right. So this is a Flash animation. And as you probably know, Flash, to my consternation, has been whatevered. I can't delete this. Why can't I delete this? Screw it. Doesn't matter. I must be on whatever. So anyway, yeah, let's talk about what receptor regulation. So here's a typical receptor, the beta adrenergic receptor. So what happens with a receptor? Well, let's back up. So the cell doesn't just respond to the receptor. As pharmacologists, we like to say it responds, or to the hormone. We like to say the cell responds to changes in concentration of the hormone. Okay, not necessarily just the fact of the hormone, but changes in its concentration. So under normal situations, there's no hormone until it comes along, it binds to the receptor and it activates something. If you have a lot of hormone and you have a constant high concentration of hormone, the cell is going to stop responding to it because it's really only responding to the change in the concentration of hormone. And then if it goes away, it's going to respond to the fact that it's gone away. So, and the reason it's doing that is because of receptor regulation. And that was probably a pretty esoteric explanation. So let's just get into the concrete of it. Basically, when you stimulate a receptor, the cell will, if you overstimulate the receptor, the cell will bring that receptor out of the membrane to kind of protect itself from overstimulation. If you're understimulating a cell, it's going to put more receptors up to try and get that stimulation back. So it's trying to find a homeostasis. And if it's getting overstimulated, brings the receptors in, understimulated, brings them up. And this is how that mechanism works. Here's your receptor, the beta adrenergic receptor, and it's got a number of sites that we'll talk about. One is this tail over here. And this is literally the carboxyl terminus of the tail of the protein. So when the hormone binds to the receptor, that changes the conformation of the receptor. And now this tail has got phosphorylation sites that become available. So it will, and it also has this activation site that will activate a G protein. So this G protein will then become active, and we'll talk about how that happens in a minute. And the alpha subunit of it will go off and help the cell make cyclic AMP. The beta subunit, beta gamma subunit, will attract GRK up to the membrane, and GRK is the G protein receptor kinase. G protein receptor kinase. GRK then is going to phosphorylate G protein receptors. Kinase, remember, make sure you know the word kinase because we talk about it a lot. Kinase is an enzyme that uses ATP to phosphorylate things. So this G protein coupled receptor, it's got ATP on it, will get brought up to the membrane by this beta subunit and then come over and put phosphates on this C terminus. Hang on a second. Okay, once this has phosphates on it, that attracts this bad boy, arrestin, up to the membrane. So arrestin will bind to those phosphorylation sites, and that's going to block the activity of the receptor. So short term, the receptor is going to be less active because arrestin is now bound to it. And the secondary thing this is going to do is receptor-mediated endocytosis. Once that arrestin is attached to this thing, it will go over into one of those coated pits. The coated pit will invaginate and bring that receptor in. So that's kind of like the mechanism of how it happens. And then over here, we can kind of talk about homeostasis in the cell, how homeostasis works. But remember the mechanism, let me just go over it again. The hormone binds to receptor becomes active, it activates a G protein. The alpha subunit goes off and does whatever it's going to do. And the beta gamma subunit stays here, brings GRK up. GRK phosphorylates the beta receptor, and then arrestin comes up. And then arrestin targets this thing to receptor-mediated endocytosis and downregulation or bringing it in anyway. Okay, so that is the mechanism of how it works. Now, how does that get balanced out in the cell? You want to think about it in terms of 4 different processes going on that affect receptors. One thing to keep in mind is you've got this subcellular population of vesicles with different proteins that can be put into the plasma membrane or not. we'll talk later in this class about glucose transporters being here. And when insulin hits the cell, it'll put that glucose transporter up in the membrane and start transporting glucose in. But in this case, we're talking about receptors being held, and they're just held here in this subcellular pool of vesicles. Now, from this pool, these receptors could be put into the membrane, okay? That's going to increase the number of receptors in the membrane, or they can be taken out of the membrane and put back into that pool. And that's what most of the cycling is all about. This is kind of like short-term cycling. Overstimulate the cell, these receptors come out of the membrane and into the pool. Understimulate the cell, and these will come back out and up into the membrane. Now you've got more receptors up there. The more longer-term control, which is always going on at the same time, is receptors from these internal pools can go, like we said in the last chapter, fuse with an endosome and then become a lysosome, and the lysosome will destroy that receptor. So if you've got long-term overstimulation of this cell, like let's say you're an opiate addict, okay? You're taking heroin every day. Your heroin receptors come out of the membrane into this subcellular pool and then over to the lysosome and get destroyed. So now you don't have receptors for opium. Take that. That'll teach you. No, it won't. You'll go back to your opium. I know you will. No, but that's kind of why, and that's why opiate users will need more and more and more in order to get high. The first time they take it, woohoo, it's fabulous. And then the second time, not as good. Now I need more to get up to that point. And so their dose increases or graduates, as we say. So it takes more and more opiates in order to get high, because they've destroyed their receptors, first internalized and then destroyed. And that's why, like, you see so much overdosing, because heroin isn't really getting them high anymore. They're only breaking even on it. So what do? Well, look, somebody's got fentanyl. Oh yeah, let's try the fentanyl. Boom. Fentanyl's so much stronger that they end up, stopping their respiration and either, being helped or dying. And then the other thing is if they go into rehab, then all these receptors come back up. They start to be made again. So once you're understimulating cells, like you stop taking heroin, you're understimulating those cells now. You've got new receptors being made by your rough ER and your Golgi. And we talked about that and how they could be put in, anterograde formation here, and they come out in secretory vesicles, and those secretory vesicles become part of this pool. And from this pool, they go up into the membrane. So you're understimulating that cell, you're making new receptors and putting them into the membrane. So this heroin junkie then, goes to rehab, doesn't have any heroin. Over the course of like a week or 2 weeks, your receptors come back up to normal. And that period is the withdrawal period. Okay, when you don't have heroin, you go into withdrawal. That's because these cells are being understimulated now because they don't have receptors to respond to it. So that's the withdrawal period. Now you've gone through withdrawal and you try heroin again, you don't know what dose you need anymore. Because back in the day, before you went into rehab, you were using, X amount. And that was because your receptors were downregulated. Now you first come out of rehab, you take a hit, boom, you're in overdose, because now the dose that used to be good for you is now way too high because your receptors have recovered. So 4 processes and 2 possibilities. You've got overstimulating the cell, which means you're going to be taking them out of the membrane and then sending them to the lysosome and destroying them, fewer and fewer receptors. Or understimulating the cell, which means you're going to start making more receptors, putting them into the pool, and from the pool into the plasma membrane. Now the cell is more sensitive or super sensitive. So there's a couple of definitions there that are important. One is desensitization. When you overstimulate the cell, it downregulates receptors and becomes desensitized. When you understimulate it, which could be by using a drug that blocks receptors, an antagonist, you increase the number of receptors in the cell, and the cell becomes super sensitive. Okay, upregulate receptors, and it becomes super sensitive.
I have a question.
Yep.
So the people who are on heroin, how come they're prescribed Suboxone when trying to wean themselves off of it?
That's a good question. So haven't talked about well, this is a good point to bring this up. When you talk about activating receptors, normally in your body, you've got an endogenous agonist. So the hormone is the endogenous agonist. Endogenous means it comes from inside. Agonist is a drug that activates the receptor. But synthetically, we can make drugs that are going to block the receptor. These are antagonists. So this is a drug that binds the same receptor site as the agonist does, but it doesn't cause the conformational change in the receptor that activates it. So it just blocks the receptor because your endogenous agonist can't get in there. So the antagonist will block whatever agonist is there. Usually you're talking about your endogenous agonist. So Suboxone is neither, it's somewhere in the middle. Okay, so the problem with heroin is it downregulates receptors. The problem with blocking them, like naloxone, will block those receptors totally. Boom, you go into withdrawal immediately, and that's pretty horrible. So Suboxone is in the middle. It activates the receptors, but not fully. It's a partial agonist. It'll activate them maybe 30% as good as heroin does. And so you're not getting high because you're only stimulating 30% of what you used to, but you're
Oh,
withdrawal.
okay.
Okay, so it's what they call a maintenance drug. It's not going to solve the problem, but it's going to stop you from having the highs and the lows. You're just going to be here, and now you can function like a human being again.
Okay, and then that's why like naloxone sprays when someone when it's inserted into someone's nose during like an opioid overdose, that's why it completely like stops it. Like that's why like shuts it down. Okay, I understand
Yep.
that better.
And
Yeah, I
I've
work in a pharmacy, so that's why I was kind of confused
Yeah,
about
yeah,
like the
yeah.
differences and stuff.
And if you ever talk to a doctor from an ER, when someone comes in on an opiate dose and they're just almost dead, you hit them
Yeah.
with Suboxone, they go from almost dead to full on withdrawal because all the receptors get blocked.
Oh, okay.
Thank you when you do that.
Thank
They get
you.
very upset. Christy, can you corroborate that?
Oh my God, don't ever give somebody the full dose of Narcan. Oh my God. The ER docs will hate you and they will kick you out of their ER.
Yeah, she's an EMT, I'm sorry.
Paramedic.
Okay, so that's receptor regulation. And the notes now have got a few paragraphs from my drug book that explains this in a little more detail, I think. Tolerance and withdrawal, blah, 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 blah. So you can read that. I think, I don't know about you, but I think it's extremely well written. Another thing I like to talk about, which, I wish I had a whiteboard here because I don't have a figure for it, is how receptors change over time for a person taking a drug like heroin or cocaine or something, where, the first time you take it, you feel really great, and then it wears off. And when it wears off, because receptors have been changed, you come down below normal. So that's your hangover, right? Even with alcohol, you get that. You overstimulate the receptors, and so they get changed, and then you come down below normal. Now, if you just sit and wait it out, you'll come back up to normal again, right? Take you a day, you'll feel better again. But if you're a trooper, you'll go back to that drug and do it again, right? So you do it again, now you're not getting stimulated as high as you were before because you've got fewer receptors. So you don't quite get to that high. And then when you come down, you're going to come down even lower because overstimulating again, fewer receptors, you're coming down even lower. So you get in this cycle of use and withdrawal, use, withdrawal, use, withdrawal, and the highs and the lows. And that's a lot of the addictive process too, especially with something like crack cocaine. Crack cocaine takes like 5 seconds to hit your brain and lasts about 5 minutes. So woohoo, woohoo, woo, and that makes it extremely addictive. Just that short cycle. A drug that lasts longer is usually a little less addictive. Okay, 11:55. Shall I go on? I can talk all day. I don't think I've taken you guys through a whole hour and 15-minute class yet. Shall we go on? Let's see. Yeah, let's go on. What the hell? Okay, so this would be lecture 2 in your notes from Moodle. And so, Gus down. What was I going to say? Lecture 2. So typically when we talk about receptors, we talk about them having three different activities or three different active domains. And this is true of most, definitely true of G protein coupled receptors, maybe a little different with some of the other ones. But in a receptor like this, G protein coupled receptor, you've got a site on the extracellular membrane where the ligand binds. And this is kind of like you hear about in enzyme kinetics in biochemistry. The ligand goes in like a lock and a key. Okay, it fits perfectly. And that perfect connection there, 1 second, is about this. Okay, this is epinephrine, norepinephrine. So this is norepinephrine, and here are the 7 transmembrane regions that all come together in the norepinephrine, the beta adrenergic receptor. So you can see as that molecule comes in, it's got 2 OHs here, and you're hydrogen bonding with 2 serines from transmembrane region 5. Okay, and then over here you've got an acid group sticking off transmembrane region 3, and that's interacting with this positively charged amine over here at the other end. So you've got, you know, this perfect connection between the amino acids in the ligand binding site and the functional groups on that small molecule. And here's a 3-dimensional view of it where you can it comes right down into the receptor. Now, what that's going to do then is, you know, maybe this transmembrane region is a little bit back, this one's a little back. When this comes in, they come forward so they can bind to that thing. And that conformational change then gets translated into all the other transmembrane regions. So you pull any part of that protein, the whole protein changes. And so when that ligand comes in and binds to the receptor, it changes conformation, and now it turns on a different or opens up another domain, which I call the activation domain over here, number 2. So this is a domain that's normally hidden, but when the ligand comes in and binds to it, it opens up, and now it interacts with a second protein, usually a G protein. So this, let's see if I have an example of that here. Okay, so here's your beta adrenergic receptor or alpha adrenergic receptor. You've got 1, 2, 3, 4, 5, 6, 7 transmembrane regions, and this long C terminus over here. So over here, this is where those serines were that bound to that end of the molecule. And then off this 3rd transmembrane region here is where that, I think it was an aspartate, which was interacting with the other end of the molecule. So that is the ligand binding site. Now, when that, when the ligand binds, it changes conformation, and then it opens up this domain over here, which is the first part of that C terminus inside the membrane. And that is going to stick into a G protein, open it up, and let its GDP come out. And then it will go over and grab a GTP, and it becomes active. So we'll talk a lot more detail about that in the next class, but this would be your guanine nucleotide release peptide. This domain here would cause the G protein to release its GDP. So number 1, ligand binding. Number 2, activation, because this is going to activate the G protein. And then the third is what we were talking about earlier with regulation of the protein. So we call that the regulatory domain. So here on this C terminus, you've got 1, 2, 3, 4, 5 sites that GRK will phosphorylate. Okay, we're going to get hyperphosphorylated here, a couple more sites over here, and that's going to cause arrestin to come up and bind to it, and then the receptor can become internalized. Also, you know, the whole idea of internalizing a receptor is, it's kind of like, depends on how long it's been exposed to to ligand. Like in the short term, arrestin will come up and block that receptor. So now it's inactive, but it stays in the plasma membrane for a while. You're overstimulating for a longer period of time, then it will become internalized. But anyway, let me back up a bit. So those are our three domains: the ligand binding domain, activation domain, and the regulatory domain. Now here's this. Here's what the G protein coupled receptor really looks like with the ligand deep inside of it. This is usually how we draw it. And I'm not sure why I put this figure up there, but it's a cool figure, I think, in my opinion. All right, and now we're getting into G proteins. I think I want to hold off on G proteins till the next class. I want to keep you on the edge of your seats. I know you're dying to hear about them. So, okay, so let's just stop sharing. I see all your faces real quick. Anthony Estevez, are you in both of my classes?
Yeah, I am.
You poor bastard. Sammy's here. Sammy's always with me. Christy, Ashley, Brian Pinello, my favorite Pinello of all time. Hunter Reynolds. I remember Reynolds. You got blonde hair, don't you? You're quiet. That's your problem. You're quiet. Coolidge. Who else can I tease? Can't tease Haley Smith because she's a little sensitive. I'm going to let her go. Oh, Hunter's hair is more brown now. All right. Well, all I'm doing is getting myself in trouble by talking too much. I'm going to let you all go. And
Hey, Dr. Slish,
yes.
are you free right after this?
Yes, I am.
Can we talk for a minute? Okay.
Who? Oh, that's Sam. Okay, I'll send you a link right after. Okay.
Yeah, perfect.
Okay. Any other comments? Okay, we'll see you next week.
See you next week.
Bye-bye.